Silent mutation. Silent mutation are those mutations in which we see that there is a change at the DNA level, but there is no change at the protein level. As a result of these type of the mutations, there is no change on the phenotype of the individual or the organism. Since the genetic code is degenerate, so there is no major effect due to the third base of the codons. So if there is a change at the third base of the codon, usually the amino acid do not change. In this diagram, we can see that what is the meaning of degeneration of the codons. There are several codons that code for a single amino acid. If we see in this diagram, that is U, UCU, UCC, UCA, UCG. All these codons, they are for the serine. Here we can see that the first two alphabets, they are same, UCC, 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 but there is a change at the third alphabet. If the third alphabet there is a change, even then the amino acid will remain same. If there is a mutation in the DNA level, the amino acid usually do not change. So this is called as degeneracy of the codons. In this example, we see that there is a change at DNA level, but there is no change at the protein level. If we read the sequence of the normal gene, and we see that here is double G, TC, TC. Instead of, as a result of the normal product of this gene, the amino acids which are produced, they will be proline, glutamic acid, cysteine, glycine. While if there is a change at the DNA level, this C is converted into T. So at the DNA level, there is a change in the nucleotide. As a result of this nucleotide, we see that the sequence of the amino acids that remain same, proline, cysteine, glycine. So there is a change at DNA level, but there is no change at the protein level. This C, which was present in the original DNA sequence, is replaced with the thymine. So silent mutations are those mutations in which there is change at DNA level, but there is no change at protein level. This is a second example of the silent mutation. In this mutation, we can see that as a result of sequence of the DNA, this is DNA sequence, this is messenger RNA which is produced from the DNA, and this is protein sequence. So there was AT in the normal sequence of the DNA, which was replaced by CG. At the DNA level, there is a change. TA is replaced by CG. But when we see the sequence of the protein, that is methionine, proline, threonine, arginine, and there is a change in the sequence of DNA, but the amino acid sequence that remains same. We see same sequence of the amino acid, methionine, proline, threonine, and arginine. We can see that there is a change at the DNA level, but there is no change at the protein level. So silent mutations are those mutations in which there is a change at the DNA level, but there is no change in the amino acid sequence of the protein.